Hello students, welcome to this lesson. In this lesson, we are going to study reproduction of bacteria, that is, asexual reproduction by binary fission. Binary fission is the primary mode of reproduction in bacteria. It is an asexual reproduction process where a single bacterial cell divides into two genetically identical daughter cells. This simple and rapid process is responsible for the exponential growth of bacteria under favorable conditions. In this process, the first step is DNA replication. The bacterial chromosome should be duplicated. This ensures each daughter cell receives a complete copy of the genetic material. The second step is cell growth. That means the cell elongates and the replicated DNA moves to opposite poles of the cell. The third step here is septum formation. A dividing wall called septum begins to form in the middle of the cell. Then the septum grows inward, guided by a protein ring. The fourth step here is cytokinesis. That means the cell membrane and cell wall fully develop at the septum and splitting the parent cell into two. The last is daughter cells. Two identical daughter cells are produced, each capable of independent growth and division. Another forms of reproduction is sexual reproduction via conjugation, transformation, and transduction. Bacteria do not reproduce sexually in the same way as higher organisms, but they can exchange genetic material through processes that mimic sexual reproduction. These processes are collectively called horizontal gene transfer and play a critical role in bacterial evolution and adaptation. So in this case, the main mechanisms include the first one is conjugation. In case of conjugation, there is a direct transfer of DNA between two bacterial cells that are temporarily joined. That means two cells of different mating types come together and genetic material is transferred from one to the other. Conjugation has been most extensively studied in the bacterium called Escherichia coli or E. coli. In the E. coli population, there are donor cells or F plus cells which donate DNA and recipient cells or F minus cells that receive DNA from the donor bacterium. F cells have a DNA sequence which is called F factors or fertility factor. These are necessary for bacterium to serve as donor during conjugation. This fertility factor supposed to contain about 20 genes and can be in the form of plasmids or part of the DNA in bacterial chromosome. So certain F genes encode sex supply, which recognizes and binds to the surface of F cells to form cytoplasmic conjugation bridge between two cells. The F plasmid replicates itself and DNA is transferred from donor to recipient bacterium through this conjugation bridge. This may also have other types of genes, including those that determine resistance to antibiotics. The second way of gene exchange is transformation. In this case, a bacterium uptake of free DNA from the environment or dead bacteria. That means bacteria take up fragments of DNA from lysed cells and incorporate them into their genome. This allows genetic variation and adaptation to new environments. This is common in bacteria like Streptococcus pneumonia. The third form of gene exchange is transduction. This is the process of transferring DNA from one bacterium to another via bacteriophage or viruses that infect bacteria. During viral replications, fragments of the host bacterial DNA are accidentally packaged into new phage particles. These phages or this bacteriophage inject the bacterial DNA into another bacterial cell. This promotes genetic diversity and can introduce new traits.